Hello everyone, my name is Jose Miguel. I am presenting here my review of the 3 scientific papers I have selected in the context of the applications topic of the Mastering Omics Data Analysis. The title I have selected for this talk is Application of Genome-wide Linkage and Association Studies to Idiopathic Pulmonary Fibrosis, MUC5B and other variants associated with susceptibility and survival. The aims of this talk are twofold. On one hand, I will present the main findings of the works by Siebold, Pelto, and North groups, where studies of susceptibility and mortality in idiopathic pulmonary fibrosis are presented. On the other hand, I will focus my talk on the omics technologies that these authors used to perform the research as examples of applied omics technologies in the quest for translational medicine. This talk will be presented following this outline. In the first place, I will do a brief introduction to the disease, then I will proceed with the methods and continue with results and discussion, and finally I will present some conclusions. In the picture, I am showing a tissue where we may observe the presence of mucus related to the expression of health-forming mucin genes within the lung and alveolar surroundings, shown as red areas. On the left panel, panel A, we see a healthy lung tissue, and in panel B to the right, a subject with idiopathic pulmonary fibrosis. So what is IPF? IPF stands for Idiopathic Pulmonary Fibrosis. And idiopathic means of unknown origin or etiology. This is a complex disease with a suspected genetic base, with almost no response to pharmacotherapy. It has a low incidence, between 4 and 30 cases per 100,000 persons a year, but with a very high mortality, comparable to those malignancies such as cancer. In the United States, there are about 40,000 persons dying every year because of IPF. So it belongs to a set of more than 100 pulmonary disorders, but it is the most severe chronic form of pulmonary fibrosis. And that is why I have selected this complex disease. It is a real public health problem. Recent research based on the application of genome-wide linkage and association studies omics have identified and described novel variants associated with an increased susceptibility and mortality in IPF. My review papers are the following ones. The ones by Mark Siebold in 2011, and Pelto in 2013, and North et al. In 2013. I have divided the methodology in the following subsections. In the first place, I will briefly introduce the different study setups. Then I will focus on genotyping technologies. After that, I will talk a little bit about the linkage and association statistical analysis basis. And then I will continue showing how the authors deal with the survival analysis. And finally, I will talk about gene expression in the lung tissue. So let's start with the study designs and samples. The review papers are very long and contain a very large supplementary material. However, I decided to present the study design by comparing them. The use of genome-wide linkage and association studies is a matter not only of having good samples from a statistical point of view, but the use is a trade-off between the expected results and costs, and of course the budgets for the projects. Siebold et al. Decided to run a prior linkage analysis to detect regions with high linkage levels in families whose pedigrees were known, in order to make a posterior fine mapping of the identified interesting chromosomic regions, as we can see in this diagram. Then, after resequencing, they expected to achieve more refined results. Complementarily, Pelto et al. Planned a cohort study using two different cohorts, the INSPIRE and the Chicago cohorts. One of the cohorts, the INSPIRE one, was used as a discovery, and the other one was used for replication purposes. Then they ran survival analysis. In contrast, North et al. Defined a more complex experimental setup combining GWAS in 3 stages with patients and data from many sources, as can be seen in the diagram I have prepared, and then combine the results from the 3 GWAS in a meta-analysis. They also performed survival analysis on cases with follow-up data on the MUC5B promoter variant discovered by Siebold et al. And several other novel variants, such as the 12 gene variants that we discuss later. So, despite the different study design that I have shown in the previous slides, common patterns of are shared among the three studies. Trade-off between cost and sample power, well-planned designs based on previous findings, and available omic technologies compatible with those costs to drive further research. In the diagram, I have tried to present the global strategy followed by CBOL, PELTO, and NOT groups. The basic idea is to search for common and rare variants if possible, using omic technologies such as genome-wide linkage and association studies to discover associations with susceptibility and mortality in IPF. Both with genome-wide linkage and association studies, genetic markers on high-density SNP arrays or regions sequenced with the help of ad hoc primers are carefully chosen to capture or tag most common haplotypes in the human populations, as we already know. So common SNPs tend to be more informative in this regard. Most of these fall outside the exons of protein coding genes, such as the one I will describe in this review, the MUC5B gene. The study outcomes are usually indirect associations based on the previous scanning with tagging SNPs. I have to say that in the three studies, genome references were used from known projects as the HapMap project and one key 1000 genome project. What is linkage? So, across meiosis cycles and generations, recombination events, depicted here in the diagram with vertical red segments, tend to produce linkage equilibrium or destroy the correlation between markers. Fortunately, markers or tag SNPs travel together during recombination events, and therefore we can study many variants following some of them, and afterwards use statistical correlations to impute the presence of other companions correlated variants. But it is not possible to sequence all variants, a specific software was used to infer haplotypes and to assess the allelic association with the disease. Multiple testing corrections, such as Bonferroni correction, was used in these studies because it is needed when we test many hypotheses to control for false positive rates. We may then compute odds ratios, assuming or not, genetic models such as additive, dominant, recessive, for the rare allele associated with IPF. In these studies, the authors checked whether the Hardy-Weber equilibrium was kept when they looked for associations. In the case of CBOL et al., they did it using the familial information, I mean the pedigrees. Because of the recombination events, markers are population dependent, and thus it is necessary to assess for the population stratification. And this is what NOT did. They got very nice results with almost no population stratification. They did a good sample selection. In the diagram, we see a lambda of about 1 between the observed versus the expected quantiles in the sample population. So they did imputation of missing genotypes and then validated the SNP by re-genotyping the GWAS results using the IPEX Gold platforms. In a GWAS with thousands of possible tests, it is of relevance to keep only statistical significant values. The threshold used here to define a genome-wide significance was 5 multiplied by 10 to the minus 8 for a typical array with about 500,000 up to 1 million SNPs directly genotyped. Again, inheritance or genetic models were assumed to compare the odds ratios of the heterozygous, that means the rare variant, and at-risk homozygous genotypes. About survival analysis, I have to say that Pelto et al. Analyzed survival incorporating into the model some covariates, among them the MMP7 blood concentration, which stands for matrix metalloproteinase 7, a protein which is present within the extracellular matrix environment in IPF patients. And Knott et al. Carry out survival analysis too. CBOL et al. Analyzed RNA samples that were reverse transcribed into DNA, while Knott et al. Got expression profiling from the ALG-RC consortium in order to study correlations between the observed variant genotypes and the gene expression in the lung tissues. Now I will present the most interesting results of the review, in my opinion. To do so, I will present some results about the variant associations, Then I will talk for a while about the effect, direction, and linkage disequilibrium. Then present the survival analysis and the effects of the variant genotype on gene expression. As I said before, Siebel et al. Found a high linkage regions in the P terminus of chromosome 11, where cell-forming mucin genes are located. I mean genes responsible for mucus formation. After discovering this region, they did a fine mapping that I have depicted in the diagram with a blue rectangle. The idea behind the LOD score is to study if an allele is transmitted more or less than we would expect by chance. Then we plot the LOD score against the chromosome position in the x-axis and study the presence or the absence of the linkage signals. Here I'm presenting a diagram with the multiple LOD score graphs for chromosomes 11, 14, and 17. Linkage disequilibrium analysis, based on the R-squared correlation coefficient, was used to assess the association among observed variants in this region of high linkage levels. In the picture we can see the P terminus of chromosome 11 in panel A, see the green rectangle, where Siebel identified a region with high linkage levels. In B panel, the fine mapping is presented within the cell-forming mucin region, and as a result, a variant in the putative promoter of MUC5B gene was identified with a genome-wide significance. North et al. Also confirmed the association of this SNP with susceptibility in IPF and found other novel variants. These novel variants, in example at the TOLIP gene loci, are not correlated with the MUC5B variants according to the observed square R low levels. That means that novel variants in TOLIP gene are independent from that of MUC5B gene. Pelto et al. Found that the promoter MUC5B variant was related with survival in IPF. The heterozygous and homozygous for the minor allele, GT and TT in these graphs, were at lower risk of mortality, despite this is the risk allele for IPF. North et al. Found dissimilar results for the variants in TOLIP. The carriers of the minor allele, G in this case, that is the protector allele with decreased susceptibility to IPF, had an increased mortality, just the opposite as observed with the MUC5B variant. Siebel and North groups also studied the MUC5B expression and its correlation with MUC5B variant genotypes. An increase in MUC5B expression was observed between cases and controls, with a factor increase of about 14. MUC5B cell-forming protein was expressed in the lung of these individuals. So let's go with the discussion. The seminal paper by Siebel et al. Showed a common variant located at the putative promoter region of MUC5B gene, which is a cell-forming or mucus-forming gene that is associated with the development of IPF. Pelto's group found that this variant in the promoter of MUC5B is also associated with an improved survival in IPF, and that the risk allele responsible for the susceptibility to IPF also confers a survival advantage to patients with IPF. So these results suggest the existence at least of two different clinical phenotypes that can be separable by the MUC5B genotype. In other words, the higher the frequency of the risk allele, the higher susceptibility to IPF, but also the higher survival in IPF. North group identified as well as the MUC5B variant in chromosome 11, novel variants in TOLIP gene in chromosome 14 and SPPL2C gene in chromosome 17. In addition, the observed effect direction of increased susceptibility and increased mortality risk in IPF for variants in MUC5B and TOLIP genes suggests that these two genes are of importance to IPS because they may modify the susceptibility on course of the disease. Susceptibility and mortality analysis were performed by genome-wide linkage and association omics technologies after considering the trade-off between cost and expected significance of results based on the power of analysis, with great results in my opinion. Then a common polymorphism in the promoter of MUC5B RS35705950 is associated with familial interstitial pneumonia and idiopathic pulmonary fibrosis. Also is significantly associated with survival in IPF. The finding of more novel variants related to susceptibility and mortality in IPF suggests that more than one gene or loci is involved in this pathology, and this result is strongly supported by the independence of variant signals found by North Group. Just to conclude, genome-wide linkage and association studies of IPF reviewed here have shown the usefulness to identify genetic variants associated with IPF susceptibility and mortality. Finally, these are the scientific papers I have reviewed to prepare this talk. The paper by Mike Sewell et al., published in New England Journal of Medicine in 2011. The paper by Ann Pelto and colleagues, published in JAMA, in the Journal of the American Medical Association in 2013, and Imrenov and colleagues, published in 2013 in the Lancet Respiratory Medicine. To prepare all this stuff, I followed the guidelines kindly provided by the professor, and that is all. I hope you like this talk, and I welcome any of your comments and feedbacks. So greetings from Tenerife Island.